Okay, this is lecture 23 prep. I'm going to divide it into shorter ones because I've been having some sort of weird technical issue, which I don't understand. Um, so probably a couple of prep videos for this topic. And we're going to talk about a little bit more about the organizer and specification, especially anterior versus posterior. We'd spent a lot of time on dorsal versus ventral, and we haven't stop talking about that quite, but we're going to look at AP as well. And then a little bit about neural induction, and not so much really, maybe I'll just put a little X through this. The patterning is very minor. We will spend a lot more time on neural tube patterning when we talk about the chick embryo. And we just simply talk about the fact that the anterior and posterior of the nervous system are patterned differently, and that the signals for those come from the mesoderm. Yes, so that's it. This is the third time I've recorded this, so I'm getting very, very good, I think, at explaining the same things over and over. These are the learning outcomes. You can have a look at those. And I'm going to actually have them in your posted notes. So we'll start by talking, just recapping. So the Spemann organizer is what we call a global organizer. So it organizes and specifies cells for both dorsal, ventral fates, and AP. So you will remember, hopefully, a couple lectures back, that where the organizer is both the dorsal axis and the posterior axis badly drawn, but that as gastrulation proceeds, the dorsal pole slides around and is more at a right angle to the anterior posterior axis. And so it's called a global organizer, though, because at the time that it's operating, it is specifying both of those types of fates. And so we've already talked about this information, so that the vegetal region is basically sending signals to specify mesoderm and to repress ectoderm in the cells immediately above. And the ectoderm, of course, has a few signals too that are repressing mesoderm and promoting ectoderm. And we looked at that in the last lecture, I think. There's always a few days between the recordings, so I can't always remember exactly what each previous one talked about. But also, as the mesoderm is specified, the vegetal region sends stronger nodal signals to the more dorsal mesoderm, so that the specification of dorsal mesoderm is specified by the vegetal region. And then inside the mesoderm, there are signals that specify ventral fates, and that includes Wnt signaling. So there's Wnt signaling kind of across the across the mesoderm, but from the organizer you have repressive, inhibitory signals that counteract that, so that we almost have an opposing gradient. So we start with dorsal being specified by Wnt, kind of early on, and then the Wnt is expressed across and gets inhibited through the organizer, and then later on we'll see how that actually leads into anterior-posterior specification. So this is a basic diagram of sort of late blastula. So gastrulation hasn't begun yet. We have this Wnt8 signal present across all of the mesoderm, and it started off with more Wnt signaling on the dorsal side and less on the ventral side earlier in development, after cortical rotation and the early part of development. Now we have it expressed in a band across the mesoderm, but the organizer, the region that is about to become the organizer, is a source of inhibitory signals. So it's going to be inhibiting Wnt. Okay. Also, BMP4, another one of these TGF-beta family members, is expressed in signaling throughout the embryo, but again, signals from the organizer are repressive or counteracting, because they don't necessarily stop expression, but they stop the signaling of the genes. For example, FrisB binds to the Wnt8 protein and prevents it from being able to signal, but it doesn't stop the protein from being there, right? It just gets in the way so that it can't signal. And some of these others, cordon, noggin, follistatin, and Cerberus, they are responsible for basically kind of in all directions away from the organizer region. They're going to counteract BMP and in some cases Wnt as well. Okay, so the ones that antagonize BMP, cordon, noggin, and follistatin. Okay, FrisB, I've explained how that antagonizes Wnt. And then Cerberus is an interesting one because it antagonizes basically all mesodermal fates, but it specifies anterior fates in the cells, the internalized cells, and especially head. So it's required for head, and it inhibits BMP, nodal related proteins, and Wnt as well. So it prevents any of these from signaling. What's interesting about all of these genes is that, or these proteins really, is that these ones and these ones are mostly needed in more, they're needed together. So if you were to make a mutation in one of these, you would see a particular phenotype, but it wouldn't get at the whole job that protein was doing, because these are all working together to not together, but at the same time, I guess is what I should have said, to antagonize BMP, for example, and the antagonism of Wnt by Cerberus and FrisB. Okay, so this is noggin expression, the little dark grains in here. And it turns out that it's exactly where the Spemann organizer is going to develop. So noggin is one of the proteins, as I said, that it basically clears the signaling for other kinds of cells, various mesoderm and other fates, and it makes these cells competent, able to be organizer cells in concert with some of those other proteins. Okay, and so here's a few experiments with noggin that tells us a little bit about what it basically does. So if you think about those animal cap experiments where you basically collect cells from the animal cap that are fated to form ectoderm, and you culture them alone, they form ciliated sort of epithelial epidermis kind of cells. And if you add noggin to those cells, it doesn't induce any mesodermal fate, so they still just sit there and behave as ectodermal cells. But if you have some cultured marginal cells, remember the marginal cells are the ones that are, if we're looking at our blastoderm embryo, you've got your endodermal cells and your ectodermal cells, and where they meet is this region called the marginal zone, and this is where all the signaling that we talked about in lecture 21 happens, that promotes the formation of mesoderm and then represses the formation of mesoderm, so you get ectodermal cells or animal cap cells forming mesoderm in a band around the center of the blastula embryo. So if you were to take some of these ventral marginal cells, and you remember that those are the ones that are able to induce ventral fates in animal cap explant experiments, if you take these, they're already specified to form mesoderm, ventral mesoderm, but it does cause the dorsalization. So that means that it's needed to specify dorsal fates, but not mesodermal fates. But once the mesoderm has been specified by other signals, nodals and some of the others that we talked about in a previous lecture, then noggin added, layered on top of that in the organizer region will cause dorsalization of that. And if noggin is expressed in the dorsal region, it can rescue UV irradiated embryos. So hopefully you remember from a previous lecture, I think it was lecture 21, that UV irradiation of the embryos inhibits microtubule polymerization, and therefore no cortical rotation, and therefore no beta-catenin in the nucleus in the dorsal region. So what you have then is a ventralized embryo. If it turns out that some of the genes that would have been expressed as a result of beta-catenin in the nucleus, it was noggin, maybe a few steps down, maybe not a direct target, but maybe eventually, then what you've kind of done is you've bypassed the need for the cortical rotation. So if the things that you've relocated as a result of cortical rotation are not relocated, but then the proteins that would have been expressed there are provided, you've kind of bypassed the problem that is caused by this UV irradiation. And so we call this rescue. And it's similar to if you have a gene mutation and you provide the protein, that's called rescue too, because now the little embryo has the protein it needs and it can go ahead and develop. In this case, what you've done is you've kind of bypassed a few steps that were that the problem that was created by the UV irradiation. You've kind of gone around it and found a way past the problem. And we refer to that as rescue. Excuse me. So that's the noggin part of the story. Cordon is a very interesting one as well, because it's it prevents the signaling of excuse me, of BMP in the dorsal region. So all the cells are making BMP, and even the dorsalmost cells are making small amounts of it. And then what cordon does is bind to the BMP and diffuse ventrally. So if you're on the dorsal side of the embryo and you've got your BMP, the cordon protein is able to bind to it and then move towards the ventral side. Once it gets to the ventral side, there is a metalloprotease, so a protease that breaks up proteins in very specific ways that will actually separate the two, so that BMP is kind of deposited back in the ventral region. And this is a way of keeping, the BMP is a diffusible signal. It can move wherever it can get to. So what you have in this sense is cordon kind of picking it up where it's not wanted and moving it back to where it is wanted, and maintaining the gradient of BMP on the ventral side, not cordon, but BMP. That's a typo. So this should remind you of something that we've talked about in a previous class before the first midterm, and just bear in mind that invertebrate and vertebrate's dorsal ventral patterning is reversed. So the dorsal nerve cord in vertebrates is a ventral nerve cord in invertebrates, and a lot of the DV patterning molecules work oppositely. And this is just a picture of it. So on the dorsal side, here's lots of molecules of cordon. You have more BMP sort of on the ventral side because of the action of cordon. Wherever cordon encounters, wherever that happens to be, a molecule of BMP, it can bind to it. And then as it's diffusing, whenever it reaches the ventral region, it could move all over the place. There's no sort of direction of it, but as it gets to the ventral side, the protease then cleaves the BMP away so that BMP is released on the ventral side. And then again, cordon is just diffusing wherever, and anytime it encounters BMP, it binds to it, and then when it finds its way to the ventral side, it gets cleaved again. This story should be quite familiar, and these are some bonus questions that I was planning to ask in the class, which I forgot to delete from these documents, so you have some warning in advance, something that you can think about. And then if you email me your answer before lecture 24 starts, then I will give probably a couple of points at least, maybe 3 points of bonus credit, because this really requires some thought. So that's just so cordon and noggin are the best studied of those genes and those proteins, so we know the most about what they're doing. So that's what we're going to focus on for that section. And just a point that organizer properties change over time. So here's quite an early gastrula, and if we take and excise the dorsal lip of the blastopore and transplant it onto the ventral side of another embryo, you get this classic result of the twinned embryo with two anterior ends and a fused posterior end. If you do the same experiment towards the end of, so here the archenteron is formed, and it's almost as far as it's going to get, and you take that dorsal lip and you transplant it onto another like early embryo, the cells that go through here first are receiving anterior specific information, and they wind up here in gastrulation. And then the next batch of cells get sort of intermediate specification. So you're not anterior or posterior, you're kind of in the middle. And then the cells that come around and enter the embryo towards the end are getting posterior information. So when you transplant the dorsal lip at that time, what you see is just an extra tail, and so a duplication of the posterior instead of the anterior. There have been experiments done midway through this process, and if you transplant some of the intermediate cells, you get something that's really hard to draw, but you do get a duplication of the trunk region, which is the this middle region between the head and the tail. And so that's evidence that as the organizer, it's not particular cells, as the cells move through and internalize and are passing this important region, the gene expression changes with time. And so the cells that are in that region and have received information are receiving different anterior versus posterior movement. Now I'm going to stop there, and I hope that this video is going to actually work, and we'll pick it up here for the second